Okay, this is the second prep for PCR. And again, if you are already quite familiar with PCR, you may not need this. But I tried to put together an interesting, hopefully interesting set of a variety of different things that we use PCR for. So why do we do it? What's the point? What kind of applications can we use? Okay, so we can start with forensic applications. And when we talk about forensics, most people are thinking about crime scenes, and identifying bodies, and figuring out who the perpetrator is. But the actual forensic term is simply about identifying an individual based um, on some unique feature, and it could include forensic analysis includes things like fingerprints and tire treads and cat hairs and things like that. There was a person who was convicted of killing his ex-wife because his cat's hairs were in her apartment, and he lied and said he had never been there, but the only way his cat's individual hairs could have gotten there was if they came on his clothes. So, there are some very dramatic things, but there are also sort of looking at populations of wildlife, especially animals like grizzly bears or, other large animals that can be dangerous. If you have places where you can collect the hair of an organism, you have these little traps with barbed wire, and they can just catch a little bit of hair as the animal goes by. You can test that individual's DNA without having to actually come in contact with the bear. So it's safer. And you can do things like maintain databases with information of different polymorphisms, so different variants that exist in the DNA of a population. You can look at things like what kind of variants are increasing in the population, what are decreasing. You can figure out who are the parents of each of the individuals that you test. You can look at the effects of, human building and expansion of human regions, and the loss of habitat and how that affects migration patterns and all sorts of things. So right from crime to wildlife, you can do this. They even use this sometimes when they're investigating things like smuggling and poaching and that sort of thing. They can identify an individual, pelt or something, and find out that that was from a protected area, and charge the person who poached that animal. We can also do it for genetic applications, and there are different ones. You can try to characterize mutations. You can check to find out there are certain sequences in some cases that have repeats that are more or fewer repeats in an individual. So the band sizes of the PCR, we run them out on a gel, and we can see them based on size with the largest pieces at the top of the gel, and then the smaller pieces lower down. And so this is the smallest piece of these, and this is the largest piece. This is something that I have used it for, is if we have chromosomes that have little deletions in them, we want to find out what exact genes are missing from that DNA. You can do PCR with primers specific for certain genes. And in this case, the wild type, there's a band for each of the genes that we are testing. The green one is one gene, the blue is another, and then the red is a third gene. And then when we do PCR with homozygous deletion chromosomes, and we get this band and this band, but not this one, then we know that this deletion removes at least one of the primer sites. So this happens to be the exact size of the deletion, but it could be only this size, or it could be only that size. So long as one or the other of the primer sites is missing, you're not going to get a band amplified. And then, if we knew that that was the missing DNA, we could then use different primers that were more focused to really hone in on exactly what sequences are present and what are not. If we have a phenotype associated with this deletion, then we know what genes are missing. We can genetically figure out, which ones are probably responsible for the phenotype. There's also all kinds of molecular applications. PCR is used in certain kinds of DNA sequencing. We can use it for probes. Those of you who've got some MBB courses in your background may know about different kinds of hybridization, southerns, northern blots, in situ hybridization. You can clone a gene, you can express the protein, and you first get the sequence by PCR amplification, and then you can use PCR to make mutations in it, and then you can test the protein. And there's a ton. There's whole entire courses on that include sections on the kinds of things you can do with PCR. So those are some of them. There's evolutionary things. You can look at genes of interest from a related species. So for example, if there was a gene that was known in one organism, say fruit flies, and I wanted to sort of find that gene in a related fly species, I would use PCR using the same primers, looking for the most conserved part, and I would reduce the temperature a little bit so that the primers could find and bind the correct sequence, allowing for some mismatches because the sequence won't be identical in the other species, and we can sometimes use that for that. They have done things like amplify DNA from Neanderthal fossils, and we have learned more about their genomes, and we have discovered that there was Homo sapiens and Neanderthal interbreeding, because most people, except people who've only lived in Africa for their whole evolutionary history, everyone has a little bit of Neanderthal DNA within their genomes. And even just sequence polymorphisms in populations, you can characterize those little variations in DNA, even if we can't see a specific phenotype associated with that. And then again, we can look at certain polymorphisms getting more or less frequent. That's an evolutionary project. There's an SFU project, or there was some years back. This is Dongya Yang, who is a professor in the archaeology department. And there were some letters that had been sealed by Amelia Earhart, who is the aviatrix who disappeared on her around the world solo flight. And he was going to use PCR for some places where people are known to be polymorphic. And they were going to compare that to the DNA they found from some bone fragments that were found on an island. Nikumaroro, that's probably not the right way to say it, is basically an island where Earhart is thought to have gone down in her plane. They think at first, it has always been thought that she just the plane went down over the ocean, and that was it, and they disappeared. But they found these bones, and it had been thought that perhaps this is where the plane crashed. It's a very remote place. However, in the end, they discovered that there wasn't a match between the DNA that we know is hers and the DNA that we thought might be hers. There were mismatches, which means that at least they were able to eliminate those bones as belonging to her. I said solo, I think I did, but it was kind of like a solo flight, but she did have a navigator. So in fact, it was a duo flight. And here's another one. Basically, the Matson lab in biology at SFU are looking at PCR-based methods to be able to distinguish certain viruses that infect blueberry plants. And so this is improving your access to good quality food. Almost everybody I know likes blueberries, and so this is a very important project. There's a lot of work around making sure that crops are safe and nutritious, and this is one of them right here at SFU. And also, if you think about the P star strain for our DMAP1 project, one of the things that Dr. Schultz did when she first got the P star stock from the Fly Stock Center was to confirm, to prove that the P element is exactly where they said it was. And so given the known location, the thought what was thought to be the known location. She designed a primer specific for DMAP1 out here, and one for the other gene on the other side. The P element is really big. It's like 9 kb or something. It might be longer. So these are not themselves going to produce a product. So if the P element is there, we use a primer that matches the inverted repeat at the end. It's called P out because it makes DNA in this direction. It's an inverted repeat, so the exact same sequence on this side makes DNA in that direction, and then the others, right, they amplify this way. And so we knew that if the P element was in the place that it was claimed, that this band should be 1500, approximately 1500 bases in length, and this one should be approximately 500. And this is what the gel looks like. Most of you have probably seen gel pictures before. This is the ladder. This is 500 base pairs. This is 1,000. This is 1,500. This is 2,000, and then it goes up from there. Or this is 1.6, 1,600. So this is a 1 kb ladder, which is a set of DNA molecules of known size so that you can figure out if the bands that you have generated are the expected size. And this is the one we expect to see from this PCR, and this is the one we expect to see from this PCR. So this was done to confirm the presence of that P element. And so just imagine with the project we're going to do that suppose we move this P element and it ends up in here. What is going to be the effect? We could still do the PCR with P out and GSP4. What would you expect to see when you look at the gel? And that's something you can put in the comment or the next steps part of your proposal. Okay, and that's it. Hopefully this was of value.